So this is the review sheet for cells and movement across the membrane, which is 1.1 in core biology. So the first thing we have to do, we have to be able to draw and label a plant and animal cell. So on the left-hand side, I have our animal cell, which is circular in shape, which is our cell membrane. In the centre, we have our nucleus. All the organelles are held in the cytoplasm, and it's where our chemical reactions take place. And then we have our mitochondria, which is needed for energy and the process of respiration. In our plant cell then, our plant cell also has those four organelles. So it's got a nucleus, has a cell membrane, our inner wall, has our cytoplasm, which holds the organelles, and it has mitochondria for energy. Also has three others. So it has a cell wall, which surrounds the cell membrane and it protects the cell. It has chloroplast, which absorbs sunlight for photosynthesis, and it has a vacuole, which stores water and cell sap. Describe how you would set up a microscope slide. So you've got a microscope slide, you place your specimen on the slide, and you would add an indicator. That should say add, I do apologise. And you will add an indicator. And the indicator then it distains the cell, so you're able to see it, because a lot of them are transparent. Then we would place a cover slip on top, and then we would place it on our stage of our microscope, and we would look under at the lowest magnification first. So how do we calculate magnification? We take the eyepiece lens and we times it by the objective lens, because there are two lenses on a microscope. Then we've got what do each of these words mean. I'm going to write it now because there wasn't a lot of space on the worksheet. So a cell are just our building blocks. And then an example would be, so for example, a red blood cell. A tissue is a group of cells working together. And my example of that would be muscle tissue. An organ is a group of tissues working together to perform a function. And then an example again, we can name any organ, heart, lung, liver, etc. And then organ system, I'm going to put at the top, so you know, a space, is a group of organs carrying out a specific function. Okay. And then what is an enzyme? An enzyme is a biological catalyst, and catalysts then speed up the rate of reactions. They speed up the rates of reactions for respiration, for example. Draw how the lock and key hypothesis works. So the lock and key theory, remember, states that one type of enzyme will bind to one specific substrate because they match like a jigsaw, and like a lock fit, and like a key fits into a lock. So we have our enzyme, and it has the active site, and the shape of our active site is our square. Our substrate then will be the same shape as our enzyme, otherwise it cannot bind, it cannot join. When they join, then they attach, they form an enzyme substrate complex. As then the reaction takes place, the enzyme breaks down the substrate into the products. Only one type of enzyme will bind with one substrate. How can enzymes be denatured? So high temperatures and extreme pH can cause an enzyme to denature. And what that means is that the enzyme will change shape and a substrate cannot bind to the enzyme. With that then, the reaction will stop. It will decrease until eventually the reaction stops. Then we have what a specialised cell is. So it's a differentiated cell, or essentially a cell adapted for a specific function. So we have different examples. We've got a sperm cell, and the main thing is it's got a tail because it needs to swim to fertilise an egg cell. We've got an egg cell, and they have a yolk which contains nutrients for a developing embryo. We've got a root hair cell, And remember, misconception, a root hair cell is a plant cell, not a hair cell for your head, a root hair cell for the roots of a plant. And with that, they've got a large surface area, they have thin walls, they also have no chloroplasts. I can add on an extra one. They don't need chloroplasts because they're underground, there is no sunlight. And they need it to absorb as much water as possible for photosynthesis. Last example is a red blood cell. It's got a large surface area and it has no nucleus. And the reason then is to absorb oxygen to carry around the body. It doesn't need a nucleus, we can carry more oxygen. What is diffusion? So diffusion is the movement of particles from high concentration to low concentration. Osmosis then is the movement of water from high concentration to low concentration. This is just shorthand for concentration. In the exam, you will need to write concentration. Do not copy me with the shorthand. Through a semi-permeable membrane. What is required for osmosis? We obviously need a concentration gradient. Then what is active transport? It's the movement of particles from low concentration to high concentration, and it's given against the concentration gradient. And then how is it different from osmosis and diffusion? Don't know why I didn't fill this out. Is that it requires energy or ATP. Okay, it's not a passive process. It requires energy or ATP. Give an example of osmosis, diffusion, and active transport. So osmosis, water for the roots. Diffusion, so oxygen, we could have put carbon dioxide. And then active transport, ions, so transport of different ions, potassium ions, magnesium ions that a plant needs, for example, in the roots. And this is your review sheet for 1.1.